Welcome to Library Seminars, a platform for the presentation of research and ideas in support of the National Oceanic and Atmospheric Administration's mission. Today's library seminar is part of the 2021 Knauss Fellows Lunch and Learn series, a monthly webinar where Knauss Fellows showcase their own research. Andrew Villeneuve, a 2021 Knauss Fellow, working in NOAA Fisheries Office of the Assistant Administrator as a Fisheries Science Interagency Arctic Policy Fellow, will be our moderator and will introduce our four speakers, each of whom will speak about their fieldwork in a lightning round of presentations. But before I turn this webinar over to our speakers, here are just a few logistical tips to help you enjoy your presentation. If you are having trouble with the audio or visual components of GoToWebinar, please log out and rejoin us. This usually resets the software and resolves most technical issues. This presentation is being recorded and will be available on the NOAA Central Library YouTube channel later today. I've added a link to our channel in the chat box. All audience members are muted, but we want to hear from you, so please ask questions. We encourage you to type them as they come to you. The questions chat box is located in the control panel. And as you enter your questions, just let us know to whom you are addressing your question by including the name of the person you'd like to answer it. All the questions will be answered at the very end of the four presentations. So with that, I'll turn the presentation over to Drew. Thank you.
Thank you, Lisa, and thank you everyone for attending today's talks. I know I'm very excited to hear what our Knauss Fellows have to present about. But once again, just stating that today's talks are a collaboration with the NOAA Library and the Knauss Fellow Lunch and Learn Committee. And as a final note, unfortunately, one of our speakers will not be able to make it today, but I know that we are thrilled to have our panel of 4 Knauss Fellows today. So today's theme is all about fieldwork and the daily life of our Knauss Fellows, which encompasses everything from engaging with decision makers and communities at international conferences to wading through marsh mud. And so with that, I'm excited to introduce our first speakers. We will actually have a split bill on our first presentation. Theresa Keith and Marina Cucuzza will be presenting on Knauss Fellow engagement in the All-Atlantic Ocean Youth Ambassador Program. So as some background of our speakers, Theresa Keith is a Knauss Marine Policy Fellow in NOAA Research, working as a UN Decade Domestic Engagement Policy Advisor to promote and coordinate participation in the UN Decade for Ocean Science. She has a master's in Water Resource Policy and Management at Oregon State and a MA in Water Cooperation and Diplomacy. She's particularly passionate about climate change, environmental education, and inclusive science, and aims to incorporate these themes in her work as an All-Atlantic Ocean Youth Ambassador for the United States. In second, Marina is a 2021 Knauss Marine Policy Fellow working as a climate and fisheries specialist with NOAA Fisheries and NOAA Research in the Office of Science and Technology Climate Program offices. Marina holds a dual Master of Science degree in Marine Biology and Marine Policy from the University of Maine, where her research focused on the resilience and management of Maine's fishing communities in the face of ecological and socioeconomic change. Marina is an All-Atlantic Ocean Youth Ambassador as well, representing the United States, where she works to educate, advocate, and engage society in efforts focused on safeguarding the Atlantic's future. I will pass the torch on over to Theresa, who will be leading off today's talks.
Thank you for the introduction, Drew. And to get started here, go back a slide. Thank you very much. So Marina and I will be focusing today, as mentioned, on the Ocean Youth Ambassador Program, taking part in this entire year so far. And to begin, I just want to give a little bit of information born out of several.
Theresa, I'm sorry to interrupt you. There's a little back sound. If you could possibly take your earphones out, it might help. I'm still hearing. Unfortunately, I'm still hearing the feedback. let's see if we can try that one more time. If you could just, do you have your phone with you? I apologize for the delay, but we're just going to switch over to the phone so that there's less feedback. Theresa, if you need any help, just let me know, and I can show you how to do that. Theresa, if you could speak, I will go ahead and.
Can you hear me okay now?
Yes, we can hear you. Great. Fantastic.
Okay, my apologies. Dialing into my phone.
Excellent. Thank you.
No problem. Okay, so to start again, thank you. Thanks, Drew, for the introduction. And I'll begin talking about the All Atlantic Ocean Youth Ambassador Initiative with some background. So first, to begin, the initiative was born out of several international agreements: the Galway Statement on All Atlantic Ocean Cooperation and the Belém Statement on Atlantic Ocean Research and Innovation Cooperation. So you can see actually these depicted geographically in the top right. One of these, the Galway Statement, was mostly focused on the North Atlantic Ocean, and the other focused greatly between South America and Africa. And so the All Atlantic Ocean Research Alliance really aims to bring these two treaties and their respective states together in an all-Atlantic pole-to-pole engagement. So it's a landmark science diplomacy initiative, and it's used to strategize joint research, to share costs for new research, and to co-develop knowledge across the entire Atlantic Ocean basin. So that's where the All Atlantic started. And a little bit about the All Atlantic Ocean Youth Ambassadors. It's the flagship program of the Alliance and an example of one of the first attempts of the Alliance to initiate international cooperation by bringing together early career researchers and early career professionals in ocean science across all of the relevant treaty countries. So the program was actually established in 2019. We had a predecessor class, and we were brought on board at the end of 2020 in December. And so, as described by the All Atlantic Ocean Research Alliance, the point is to enable the next generation to become actors of change while promoting stewardship of the sustainable development of the Atlantic Ocean. And the mission statement really is to empower youth both through career development, but also empowering them, giving them a platform to engage with citizens from our respective countries about science, about the ocean, and also to share across borders different things that we're doing to promote a cross-ocean, cross-Atlantic initiatives for sustainable development, as well as for ocean literacy. So a little bit about us. You can see all of our faces there in the little bubble. We were officially inducted in February 2021, and there are 25 of us spanning across 14 different countries, and we have a range of backgrounds. So Marina and I, of course, are Knauss Fellows. We have some counterparts who are in master's or PhD programs in oceanography, in microbiology, etc. We have some colleagues as well who are in filmmaking and ocean literacy initiatives, some who work for nonprofits, and some who work for government agencies as well in different countries. The All Atlantic Ocean Youth Ambassadors come from a range of backgrounds, and that range really gives us a broad set of skills to put together different initiatives. So people who are great at making videos or great at making social media posts. And together we come up with different creative ways to engage people in our own countries and also around the world in development and stewardship of the Atlantic Ocean. And so our role really is to be a sort of a figurehead group, a flagship group showcasing what international collaboration can look like and also showcasing this shared interest across all of these different countries and across our generation for sustainable development, sustainable use of the ocean. So what are we actually charged with doing? We do a number of different things to promote change for the Atlantic Ocean. Some of those things include developing campaigns and communication strategies, which Marina will talk about a little bit later. Of course, what we do to encourage and inspire is a little bit different in the digital age from what our predecessors were doing in person. But we have been making the most of different platforms virtually to engage people both in our own communities and also at different international treaty meetings, at conferences, and also through our own series of webinars, which Marina will talk about as well. So it includes reaching out to people in our own communities and our respective countries, students at our own universities, civil society, decision makers, all of the people who are engaged in these relevant treaties from our different countries. And our focus is really on conservation and protecting the Atlantic Ocean, and of course all oceans since they're connected, and really encouraging early career researchers and early career professionals to be engaged going forward. So the three words we like to use to describe our actions are sharing, sharing information about our own research, about the All Atlantic Initiative, and about the ocean in general, and promoting ocean literacy, connecting both to our own communities as well as to each other across the ocean, and acting, encouraging people on the ground to do things, from beach cleanups to different initiatives to promote ocean literacy and also to promote sustainable use in their own communities and in things like fishing and things like use of different resources. So here in the picture you can see a few of the different activities, and Marina will talk more in detail about what we have been doing this year. And now I'll pass it over to Marina to talk about some of our own flagship initiatives and how we've been engaging so far.
Thanks, Theresa. Good afternoon, everyone. So now that you have some background on the Ambassador Program, I'll be highlighting some of the activities we've been able to engage in so far this year as part of the program, starting with the All Atlantic Research and Innovation for a Sustainable Ocean. So this was the ministerial high-level and stakeholder conference that was put on by the Alliance, held June 2nd through 4th. And the hosts were the Portuguese presidency of the European Union Council and the European Commission. The theme of the conference was around connecting, acting, and cooperating. So the conference brought together high-level ministries, as well as academics, stakeholders, civil society, and youth to discuss research and innovation happening throughout the Atlantic. Ambassadors were able to participate in a number of different events throughout the conference, such as discussions on the R&I Solutions pledging platform that was launched. A few ambassadors were able to engage in a debate on rethinking our ocean stewardship, which talked about best practices to bridge the emotional gap between society and ocean conservation challenges. The Portuguese ambassador was able to attend the conference in person, and she helped to moderate an event on a plastic pollution campaign that's being launched in the Azores. And finally, we had an ambassador speak about the Joint Pilot Actions Plenary, talking about the Ambassador Program. So at the All Atlantic R&I for a Sustainable Ocean Conference, the ambassadors were able to host our own side event, which was a panel focused on opening doors for early career ocean professionals. So for this panel, we invited 5 speakers from different countries, all with really diverse backgrounds in ocean science, all in very different stages of their career. And we invited the audience to ask questions, and together we talked about barriers facing youth in ocean professions and discussed possible solutions. So some of the major topics that came up during this panel discussion include different career options that are available for people interested in marine science, how mid and late career professionals can help to be allies to early career professionals. We also touched on youth empowerment and diversity and inclusion in marine sciences. And in total, there were 15 different countries in attendance, and it was a really lively discussion. So a big part of, thank you, Theresa. Next one, perfect. So ambassadors were also able to attend Virtual Early Career Ocean Professionals Day, or VCOPS Day. So this was a 24-hour live stream event that follows the sun around the world. And it was hosted immediately after the first international conference on the UN Ocean Decade for Sustainable Development. And VCOPS Day is hosted by and for early career ocean professionals, and it's intended to serve as a platform to really showcase their work, activities, and contributions to the UN Ocean Decade. So ambassadors from the US, Canada, and Brazil put together a video highlighting the contributions that our countries are making towards the Ocean Decade. And for our portion of the video, Theresa and I were able to interview Nancy Knowlton, who's on the US Decade National Committee, and we asked her about perspectives on the Ocean Decade, and she talked about opportunities and challenges that she saw related to the decade outcomes. Let's see if I can get the next slide up. Here we go. Thank you. So a large part of what we do as ambassadors is we launch social media campaigns. And these campaigns are really intended to inspire and to highlight positive changes that are occurring along and across the Atlantic. And these campaigns are focused on ocean literacy, community engagement, and research. So here's some examples of some of our posts to highlight these different campaigns. For example, on the top left, here's a series of posts we were creating leading up to Earth Day, highlighting climate impacts on the ocean. So each day we would post these short little vignettes with research and short pictures for people to click on for more information. On the top right, here's a group of our South African ambassadors who developed their own beach cleanup, and they launched that on Earth Day. So this is an example of the type of community engagement activities that many of the youth ambassadors are participating in that we post on our social media. Bottom left is a series of campaigns leading up to World Water Day, and we asked ambassadors to share posts and start conversations around water and the importance of water to livelihoods, well-being, and the environment. And finally, on the bottom right, that's Kelsey, who's one of the ambassadors from the UK, and she's currently out at sea on a research cruise as part of the Research Alliance. And she's been doing a lot of really awesome live streaming of the work they're doing at sea to do some different science communication activities as well. Next slide, please. So I'll end by just talking about some next steps for the ambassadors. We'll be consulting and planning for the next All-Atlantic Research Alliance Forum, and we'll also be developing and expanding our outreach campaigns. So if you're interested in staying in touch and following the work that we do, we post actively on Facebook, Twitter, and Instagram, and we also share a YouTube account with the All-Atlantic Ocean Research Alliance. And if you go to that YouTube account, you'll find videos from our VCOPS Day and also our panel on opening doors for early career ocean professionals. So feel free to stay in touch and follow us. And last slide is just thank you very much for listening, and we're happy to answer questions after the rest of the lightning talks.
Okay, thank you, Marina and Theresa, for that awesome talk. I really enjoyed learning a bit about what you were doing in your Knauss year. It's always great to see other fellows and their work. Up next, we have Max Showalter, who is an Interagency Ocean Policy Fellow with the NOAA Ocean Exploration and Research. So with a love for all things cold, Max Showalter earned his PhD in oceanography and astrobiology at the University of Washington, studying microbial communities within sea ice. His research focused on understanding how bacteria and their viruses survive extremes of temperature and salinity in the Arctic as an analog for icy moons, as well as work understanding Inuit environmental policy in the Canadian Arctic. And so today, Max will be presenting Finding Life When the Trail Goes Cold, Microbes and Ice from Sea to Space. Max, take it away.
Thanks very much, Drew, and thanks everybody for giving me the opportunity to talk to you today. A little bit about some of the work that I did during my dissertation, which was, like Drew mentioned, focused on understanding life within sea ice and how that can help us understand a little bit more about how we look for life in outer space. And so first, before I start, I just want to acknowledge the folks who funded this research and some other people who were involved in what I'm going to talk about today. So my goal for this short talk is to ensure that as you leave this talk, you'll be able to understand a little bit about what astrobiology is and why it's important. And as part of that, I want to talk to you about why sea ice is one of the best places on Earth for us to ask some of these really important questions about astrobiology. And the reason that sea ice is so important is because we have bacteria and other microbes that survive within these really extreme conditions in sea ice. And that helps us understand more about extreme life in outer space. So these are our goals for our 10 minutes together today. So I want to jump right in and just offer this really simple definition of what astrobiology is. And so astrobiology is just simply the study of life in our universe, which of course is this really huge topic. And so we can break it down into some fundamental questions that help us approach this broader research goal. And so the first question that we might ask as astrobiologists is, how do we find life? And so an example of this is, what are biosignatures that we can look for in outer space that indicate life might be there? And so a lot of this work, for example, is performed by astronomers and geologists. So the former Kepler mission at NASA helped us identify a lot of exoplanets beyond our galaxy or beyond our solar system that might host life. Another key question would be, how did life originate? So if we're looking for life in outer space, it helps us figure out where to look if we know what are the conditions that allow life to arise and evolve. And this is actually work that a lot of folks at NOAA support. So for example, from my home office, NOAA Ocean Exploration, with investigating things like hydrothermal vents, where we think life might have originated on Earth. And then a third key question in astrobiology is asking, how does life survive at extremes? What are the limits of life? So you can imagine if we're looking for life in our solar system or the rest of outer space, if we have a good understanding of the limits of life, that helps us sort of define where we should be looking first. So here's a picture, for example, that some of you might recognize of Grand Prismatic Spring at Yellowstone National Park. And all the bright colors that you see here are actually archaea and bacteria surviving at really extreme temperatures and pH conditions. And this is what I'm going to talk to you a little bit about today. This is what is most connected to my work. And you can imagine when we're studying how life survives at these extreme conditions, or when we want to identify what are the limits of life, we have to look to the only example of life we have so far in our universe, which is life here on Earth. And so we look at some of the most extreme places on Earth. One of these places is sea ice, and sea ice is a really great analog for outer space environments because it is one of the most extreme habitats on Earth. So obviously sea ice is very cold with temperatures down to -40 degrees Celsius, but it also experiences intense seasonality and really high salinity. And let me tell you a little bit about what I mean by that. So here's an image of sea ice at the North Pole, and if you were to take a cross section of some of this ice and zoom in on the micrometer scale, what you'd see are all these little liquid pockets. So here, this is as a cross section of some ice, and you can see these little pockets in the ice, which are known as brine pores. So brine pores are the result of how seawater freezes. As the seawater freezes, the salts inside seawater get trapped inside the ice matrix and then concentrated as the ice grows. And this allows this liquid network within the ice to persist, even though the temperatures are below freezing. So what you end up with is this matrix of liquid pockets that are low temperature and very high salinity. But because they're liquid, they can still host microbial communities. So this is a really good place for us to study life in outer space because it's such an extreme environment. And one of the questions, of course, we can ask is, how do organisms survive under these extreme conditions of low temperature and high salinity, like we see in sea ice brines? And so this was one of the questions that I tried to answer, or at least answer part of, with my dissertation. And in particular, I was focused on understanding how bacteria access the nutrients or the food that they need in order to survive in these conditions. And so I want to introduce you to extracellular enzymes, which are an aspect of that question. So enzymes, of course, you're all familiar with, are just proteins that help initiate or catalyze reactions. Extracellular enzymes are doing the same job, but they do it outside of the cell. And extracellular enzymes are really important because most organic material, most of the food that cells need, is too big to be consumed without being broken down first. And so that's where enzymes come in. So if you look at this schematic here, we have a cell over here on the left. This little green blob is an enzyme. And so what happens is the cell will make the enzyme, release it into the environment where it interacts with one of these large substrates, so something that's too big for the cell to eat. But after the enzyme interacts with it, we have these little bite-sized pieces that the cell can then take up to fuel its metabolism. So you can imagine, because most of the material is too big to be taken up without this digestion, extracellular enzyme activity is like the rate limiting step for a lot of carbon cycling and organic matter cycling within the environment. So it's really important. And we also know that enzymes have a really hard time operating under extreme conditions like low temperature or high salt conditions like we have inside of sea ice. So we can ask a few key questions here. First, are extracellular enzymes actually active in sea ice? So we know it's a challenging environment for them. Can they actually do their job? And if they can, how does that rate of activity in ice compare to the underlying water? And then finally, what are the limits of extracellular enzyme activity? If we're looking for life in outer space, we want to know what are the temperature or salinity conditions that we can sort of draw the line and say there's no need to look beyond this point. So to answer some of these questions, we have this really elegant assay that uses fluorescence to measure enzyme activity. So let's say we have some substrate which is artificially produced. So that's these three blue balls right here. So this is analogous to that organic matter that we'd see in the environment, but we tagged it with a little fluorescent tag. And so what happens is when we add that artificial substrate into a sample, the enzyme that we want to measure the rate of comes along, interacts with the substrate, and it cleaves off that fluorescent tag, which then glows. And so we can measure the change in fluorescence over time to get a rate of enzyme activity. So this was the fundamental method that we used for this project. And we wanted to do this on ice samples and the underlying water. So we went up to the North Pole. Here's a picture of the ship we were on, the Swedish icebreaker Odin, moored to this giant ice floe here at the North Pole. We affectionately dubbed it Big Bubba. And what we did with Bubba is moor to the ice floe. Here's a picture of our cruise track in light pink. And then what you see in dark red here, we moored to the floe and then just drifted with it for a couple of weeks. And so as part of this research, we wanted to look into the ice. So we had to get ice cores. So you can see that's what we're doing here. We have a giant core that we take into the ice, and that's what all of these holes are. You can see the ice coring can be very hot work. So here I am in a t-shirt at the North Pole. So we core into the ice, and then we were most interested in the bottom part of the ice where most of the bacteria and algae are. So I'm going to walk you through just some results from this research. And so what I have over here on the left is a plot of some of the enzyme activities that we measured at the North Pole. So here are three different sample types. The first two on the left are sea ice, and then this over here on the right is seawater. And the y-axis is measuring the rate of enzyme activity normalized to the cells. So this is an indicator of how active the enzymes are in this environment. And so what you can see is that we do have enzyme activity in these sea ice samples. So that sort of answers our first question. Yes, the enzymes are active in these sea ice samples. And when you compare it to the seawater sample over here on the right, you can see that the sea ice samples actually had higher activity than what we saw in the underlying seawater. I've also included here the temperature and salinity conditions for each of these samples. And so you can see here in the center, we had sea ice and we measured it at -10 degrees Celsius and 142 parts per thousand salts. So about 4 to 5 times the salinity of seawater. And that's actually a new low temperature, high salinity record for enzyme activity. So what we were able to see is that enzymes are active in sea ice more so than in the underlying seawater and to very extreme conditions. And this work is actually going to be published next week. So if you're interested to learn more about it, I've included the DOI in case you want to look later. Okay, so we had our key research questions. Are enzymes active in sea ice? The answer is yes. How does enzyme activity compare in ice to the underlying water? Well, we saw it was higher in the ice. And we didn't reach a limit of extracellular enzyme activity, but that in and of itself is information, right? We know that the enzymes are active down to these very extreme conditions. So this is really important because enzymes help us understand life both on Earth and in outer space. So here's a picture of Enceladus, which is an icy moon of Saturn, where we think we might be able to find life in this liquid ocean and icy crust on top. And when we think about this information, first of all, it helps us understand the carbon cycle on Earth a little bit better. So I mentioned earlier that enzyme activity is sort of the rate limiting step for a lot of carbon and organic matter cycling. And in the Arctic and also the Antarctic, this is sort of a black box. We don't know how carbon gets cycled, especially under extreme conditions. So this adds information to that. This also helps us understand and expand our definition of what habitable space is. We can now say that enzymes and correspondingly the cells can be active at these very low temperature and high salinity conditions like we might find on places like Enceladus. And because this assay is so simple and detects low levels of cells, it could be a useful biosignature when we're out in space looking for ways to detect life. So just to summarize, today we talked a little bit about astrobiology, which is just the study of life in the universe, where it came from, how it evolves, and where we can find it. And we showed that sea ice, because it's such an extreme environment in terms of temperature and salinity, is a really good place for us to look at these questions on Earth. And the reason that sea ice is such a good analog for astrobiology is because we already have these microbial communities in sea ice that survive under these really extreme conditions of low temperature and high salinity. So I'm going to wrap up there. Thanks very much, Drew, for the opportunity to talk today.
Great, thank you, Max. That was super interesting. I very much enjoyed learning about those extremophile bacteria. So up next, we have Lu Wang, who is a Knauss Fellow with NOAA Ocean Exploration. And so Lu began her science career studying rangeland ecology and plant biology, including a study on the effects of various watering methods on the microbes living on agricultural plants. This interest in plant microbe interactions led to her PhD work on the influence of environmental perturbations on microbes living in seagrasses. Studying seagrass ecosystems and sediment biogeochemical cycling sparked an appreciation of coasts and oceans, leading to her current position as a Knauss Fellow with NOAA Ocean Exploration. The title of her talk today is Adventures in the Field as a Seagrass Microbiologist. And Lu, I will go ahead and hand it over to you.
Great, thanks, Drew. Thanks for that introduction. And thank you to the Knauss Lunch and Learn Committee, as well as the NOAA Central Library for having me. So today I'll be speaking a bit about the fieldwork aspect of my graduate work, which focused on seagrass microbiomes. So first, I want to introduce seagrasses. So these are plants that live in the ocean. They can be found along coastlines and estuaries globally. I actually took this photo in Newport, Oregon, where I did most of my research. And seagrasses are really important. They're highly productive ecosystems. They provide a food source and habitat for coastal fauna. Their root structures can prevent coastal erosion. And importantly, they can also sequester carbon within their sediments. And so impacts on seagrass meadows can result in impacts on other species at the ecosystem level. So for my graduate work, I mainly focused on seagrass microbiology. So seagrasses, they host unique microbial communities. So these are different from the microbiomes of the seawater or the bulk sediment. And this makes sense because different parts of the seagrass, so the leaves and the roots, they present unique environments in terms of oxygen concentration or resource availability. So for example, carbon, and these conditions allow for a certain group of microbes to thrive. And so from this interaction, the microbes can benefit. Again, there might be extra food, carbon for the microbes left over from plant photosynthesis. And in return, the microbes can help the plant out by consuming phytotoxins, creating nutrients for the plant to take up, and even acting as competitors against plant pathogens as the microbes are physically competing for space on the plant. So for my dissertation, my main question was, how do environmental changes affect the seagrass microbiome? And in turn, do these changes in the microbiome have the potential to affect seagrass health and ecosystem services? And so as part of my dissertation, I explored the effects of 3 environmental perturbations on the seagrass microbiome. Nutrient pollution, so that's kind of asking the question of what happens with fertilizer runoff. The effects of transplantation on the seagrass microbiome. So as seagrass populations are declining, a way to restore these populations would be through transplantation. But how does transplantation affect the seagrass root microbiome? And a third part of my dissertation looked at the effects of ocean acidification on these plant bacteria. So a typical protocol for what I followed for my work, which mainly looked at 16S, amplicon, and metagenome data, you start out by obtaining samples of interest. And so for me, that would be a seagrass leaf or a root sample. You extract DNA from the surface of that leaf or root to get the microbial DNA. You prepare that DNA for sequencing, send it off to get sequenced, and then process and analyze that sequencing data when you get it back. Unlike most of my talks about my dissertation, this talk will mainly focus on how I obtained the samples of interest. Since I mostly looked at the effects of environmental perturbations on seagrasses, I had to create many of these perturbations myself. So I'll be highlighting how I conducted these experiments, mainly focusing on the nutrient pollution experiment and the transplantation experiment. So step one would be getting sediments. Seagrasses are plants, so they're best grown in sediment or soil. So in this photo, you can see me and my lab mate helping me out as I try and shovel sediment into a bucket. This is, I think, while the tide is coming back in, which is not recommended, but we still made it work. Step two, after you get the sediment, you want to, or still part of step one, you want to transport that buckets of sediment back to our workspace. Most of this work was done in Newport, Oregon at the Hatfield Marine Science Center. So we put these sediment into these 7-gallon buckets, as you can see here, which turned out to actually be really heavy. You can see that it's taking 2 people to transport this bucket. I will also say that it was really difficult to maneuver these heavy buckets across mud flats, because when you're walking through a mud flat, you're constantly sinking in the mud. So imagine doing that while you're carrying like a 100-pound bucket. And so this was definitely a really big production, and we eventually learned to just fill the buckets halfway. And lastly, once you have the sediment, you want to transport that back and begin filtering. So we filtered the sediment with mesh wiring, and the reason we did this was to remove large debris, things like shell pieces, clams, crabs. If we found any living organisms, we returned them back to the bay. And this was also a multi-person, multi-hour effort. We were putting these filtered sediments back and forth in the fresh buckets, just kind of a mess everywhere. So after we got our sediment prepared, we went back out and got seagrass. So again, go back out during low tide in our waders and boots. Whenever we did this, we would always bring bags and buckets and fill that with seawater, and that's where we would put all of our collected seagrass. That's actually one of the first rule of seagrass fieldwork: don't let your seagrass dry out. Again, that is my lab mate helping me out, along with my PhD advisor, Dr. Ryan Mueller. This would take a very long time if I was doing it by myself. So step 3 of all of this would be to create your perturbations. And so for my nutrient pollution work, I planted the seagrasses in separate tanks. And the tanks were randomized into fertilized or control samples. And so half of the tanks were introduced with slow release fertilizer, and the other half were left as controls. And so in this slide here, you can see the photo on the left would be all of my tanks. And this photo on the right was actually taken from a different experiment, but same idea, where you introduce slow release fertilizer by wrapping it up into mesh and then stuffing that mesh bag into a PVC pipe that we cut up. We zip tied that PVC onto a stick and then introduced that to the tank. And then for our ambient tanks, we would have the same device in the tanks, but just without the fertilizer. So for the transplantation experiments, so our question was, what would happen to the seagrass root bacteria if we planted them with or without the sediment around the roots, which is known as the rhizosphere? And so in this drawing here in the middle of the slide, you can kind of see our planting scheme, where the drawing of the seagrass on the left shows a seagrass plant with bare roots, and the drawing on the right here shows a seagrass plant with sediment attached, or as much sediment of the original sediment that it came with. And then we cut up PVC pipes again, stuffed it with the sediment that we had filtered, and then we planted each seagrass plant into its own PVC tube. And after we planted all that, we put the PVC tubes full of plants into a giant tank with flow through seawater. And so this way, whenever we would sample, we would randomly pick a PVC tube to sample. So this whole design was to make sampling easier, because we wanted to look at the rhizosphere. And so if we just picked it up, without the PVC tube, the rhizosphere would just kind of disintegrate into the water column. And lastly, I wanted to touch upon another chapter of my PhD, which centered on how seagrass microbiomes would be affected by ocean acidification. And so for this project, instead of actually simulating acidified conditions, samples were taken off of Ischia, Italy, which is shown on this map here. And so in this area, there are naturally occurring CO2 events that kind of mimic the effects of ocean acidification. And so in this picture on the right, these bubbles actually show the CO2 coming out. And these seagrass meadows, they've been here for over 10 years. And so this really shows how the system can adapt to a perturbed environment over time. But unfortunately, I didn't get to go there, and my PhD advisor and co-authors took a boat out and dove down to get the seagrass samples. I just wanted to highlight some key conclusions from my dissertation. So I looked at 3 environmental perturbations on seagrass microbiomes, nutrient pollution, transplantation, and ocean acidification. All 3 of these perturbations actually did end up eliciting plant changes, plant responses, either in the amount of carbon that is available to their microbial communities, or potentially changes in the recruitment of microbiota. This is especially the case in the transplantation study. And so all of these plant responses in turn resulted in changes in the microbiome, either functionally or in composition, and changes in the microbial communities in this area can ultimately lead to ecosystem impacts. And so that would be potential shifts in biogeochemical cycling. These microbes are especially they play a big role in carbon, nitrogen, sulfur cycling, and these changes can also go back and have impacts on the host health and the general habitat suitability. I also wanted to touch upon lessons that I learned outside of microbiology and things I didn't expect to learn through this graduate experience. I learned that a 7-gallon bucket full of sediment is really heavy, and I actually can't lift it. I've learned how to grow seagrass. That was something I was really worried about going into my PhD, just worried about whether I could keep seagrass alive for long enough to do an experiment. I learned how to use power tools to cut PVC pipes. As you can see from this talk, my lab loves to use PVC pipes. And I also got really good at walking through mud flat towards the end of my PhD. To conclude, I just want to say thank you to everyone who provided field assistance and funding over the years, especially to my PhD advisor, Dr. Ryan Mueller, as well as the Hatfield Marine Science Center in Newport, Oregon. Of course, thanks to the Knauss Fellowship, without which I wouldn't be here. Thank you all for listening. And if you want to contact me or follow up about anything, there's my email. Thanks.
That was a lot of fun. I had such a good time listening to all the fieldwork stories. And this is our Q&A time, so I want to invite all the fellows to pop back up on their webcams. And audience, we have about 15 minutes to answer your questions. So this is a great time to type them into the questions chat box in the control panel. Make sure to let us know if you're addressing a particular person so that we can direct that question correctly. So it does take a minute to get the questions. And while we're waiting, I just want to also encourage audience members to take a look at the handouts drop-down menu that's also in the toolbox. Some of our speakers have shared their slides, so if you want to take a closer look, since these, of course, were lightning talks, please feel free to download those slides. And of course, we are recording this webinar, so if you heard a presentation and you think someone you know might want to hear a little bit about this, please share the link to the webinar with a colleague. I'm going to put this webinar on the NOAA Central Library YouTube channel later this afternoon, probably in about 2 hours. So that link again is in the chat box if you want it. So we just got our first question, and it's directed to Lou. It asks, how long did you let the seagrass acclimate before starting your experiments?
Yeah, that's a great question. I believe, depending on the experiment, anywhere from 2 weeks to a month. So we definitely let them settle before we did anything to them.
Great, that's wonderful. This next question, I don't see who it's to, but I'm going to read it out loud. What kind of opportunities are available to law students through the Knauss Fellowship? I don't know if anyone can field that. That might be a question that we would have to answer offline, and to the person who asked that question, I'll be happy to forward to the Knauss group so that we can get more information about that. I do have another question here from Max. It asks, are the bacteria found in the brine channels obligated to that environment, or can they also be found in the surrounding seawater?
That's a great question. So most of the organisms that we see within sea ice brines are also found within the surrounding seawater, because if you think about it, sea ice, and increasingly more so, only lasts for 1 year. And so the organisms are sourced from the seawater that freezes to become the sea ice. But interestingly, we see a lot of the same sea ice organisms surviving in the Arctic and the Antarctic, and also in a lot of deep sea environments. So for example, after the 2010 BP Horizon oil spill, they investigated the response to the deep sediment, or to the benthic microbial response to the oil spill, and they saw a lot of the same organisms that we see in sea ice. So they're spread all over, but have unique traits to survive in the ice.
Thank you, Max. So I'm still waiting for some questions to come in. I'm actually curious, Theresa and Marina, how does somebody get involved in the ambassador program that you were part of?
Theresa, do you want to start?
Sure, yeah. So as you mentioned, there's a number of different countries, and so it's kind of country dependent. In our case, we were recruited through the Knauss Fellowship program by filling out an application, but it depends on the country. So in the UK, for example, or in South Africa, usually the institution that's involved in managing either the Galway or the Lem statement involvement for that country, they're in charge of putting out the call to people in their own career ocean professional community in different ways. So it's kind of different per different countries depending on which institution is involved in enacting the treaty.
And I'll just add that some of the ambassadors from last year have been able to stay on and help be mentors to us too. So it's been nice to have them and learn about their experience and have them stay on this year to help us as well.
Thank you. I appreciate your kind of clarifying that. I was very curious about that program. Another question just came in. Also, it's to Max. It says, do some of the bacteria cheat? For example, do some let other microbes in the brine pool to use resources to create extracellular enzymes and then reap the benefits of the broken up food?
Yeah, that's a very astute question. We expect the answer is yes, that there are a lot of cheaters in brine pores that don't produce these enzymes, but still get all the benefit of it. And this is a known phenomenon that we see with extracellular enzymes in other environments. But we think it might be especially important within sea ice brines for a few reasons. So first of all, the brines are often discrete. So you just have this small environment that lasts for a couple of months. So if you have somebody producing enzymes, they can accumulate within that environment. And then all the other cells can still get the benefit even if they're not producing them, because they're all tightly, spatially co-located. Another reason to expect that it might be significant in sea ice brines is because there's lots of really goopy organic material there produced by algae. So exopolysaccharides or exopolymeric substances, what they call their EPS. The algae produce these. It's basically like algal snot, but it's really good food for the bacteria. And so if they can produce a lot of enzymes, they have this nice source of food throughout the winter that they might be able to use within this environment. So we suspect a lot of them are cheaters in this environment. We don't know for sure, but the answer is probably yes. That's a good question.
All right. This next question is for Theresa and Marina. Are the positions you have for AAORA a paid job, or is it more like serving on a committee? How are you supported?
So these are all volunteer positions. So all of us are volunteers, and we're doing this as part of, on top of our work and our fellowships. So I think that's definitely a challenge, is just finding time to coordinate when this is sort of a side job for many of us, and we all live in different countries. So that's definitely been a challenge we've run up against. But Theresa and I are supported through our Knauss Fellowship through the International Affairs Office.
Thank you. So I'm going to get, oh, here's another question. This one is for Lou. When looking at the effects of ocean acidification on seagrass microbiomes, did you compare the microbiomes between samples taken from Oregon and Italy, assuming that Oregon is the non-OA site?
Yeah, so controls, important. So the species that I studied in Oregon is Zostera marina, whereas the species I studied from the Italy site was Posidonia oceanica, so completely different. And so my controls for the OA study were actually taken from a nearby site in Italy that was far away from the vents, but still had the seagrass meadows.
Wonderful, thank you. And I'm going to give it another second to see if we get another question. Meanwhile, did any of you have any last comments that you wanted to add? I know that we gave you so little time to talk about such enormous adventures and research, so if you do, this is a good time to let people know any additional information. All right, so it looks like there are no more questions. So I'm going to go ahead and conclude today's webinar, and I'd like to thank you all for sharing your fieldwork adventures with us, as well as Drew for being an extraordinary moderator, and the Knauss Fellows webinar committee for organizing this series, which helps showcase some of our young, exciting professionals here at NOAA. Drew, do you have any last comments?
Yes, I just wanted to announce that we will be holding one more, sorry, not one more, we'll be holding a couple more, but the next Knauss Fellow Lunch and Learn series of presentations will be on September 16th at noon Eastern time. And thank you everyone for coming out, and thank you to our wonderful presenters.
Absolutely. Well, I just would like to say that NOAA Central Library is very proud to host the presentations like today's that feature the work of the Knauss Fellows and the NOAA community, and that we hope you'll join us again in September. Take care, everybody. Bye.